watching Steven Loves Science. Now this episode is the first in a series of episodes covering the generation of ATP from glucose. Now, as hopefully we all know, the breakdown of carbs, glucose specifically, is a major source of cellular energy, and the complete oxidative breakdown of glucose can be written as follows. C6, H12, O6, plus 6O2 yields 6H2O and 6CO2. Now, this reaction produces a lot of energy, and it has a Gibbs number, or an energy change, of negative 686. So if you saw my video on free energy, you should know that this is a lot of energy. Okay? So now in order to harness all of this free energy in a usable form, glucose is oxidized within cells in a series of steps coupled to synthesize ATP. But we must start somewhere. So this first step is known as glycolysis. The reaction of glycolysis leaves us, like we'll see over there, with 2 pyruvate and 2 molecules of pyruvate and 2 molecules of ATP. Okay, so here we have the initial pathway of glycolysis, and now in this initial path we are actually consuming energy. So here we use ATP to phosphorylate glucose. So by phosphorylate, I mean ATP is going to lose a phosphate, so ATP is going to go to ADP, adenosine diphosphate, and we're going to donate one of those phosphates to glucose to yield glucose 6-phosphate. And just to briefly go over some of the nomenclature, the reason why it's glucose 6-phosphate, not glucose 5-phosphate, is because the phosphate group is located on the 6th carbon of the chain. So the way organic chemists do this is you start with the first carbon, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6, and that's where we have the phosphate. So that's where the substituent in this case is the phosphate. Okay? And in addition, we're also phosphorylating fructose 6-phosphate into fructose 1,6-biphosphate. And just to briefly go out on a tangent, the enzymes that catalyze these two reactions, hexokinase for glucose and phosphofructokinase for fructose, are both very important regulatory factors in glycolysis. The key control element is phosphofructokinase, which is inhibited by high levels of ATP. So inhibition of phosphofructokinase subsequently results in the buildup of glucose 6-phosphate, which in turn inhibits hectokinase. So that basically stops the process. So I just thought it was interesting as an evolutionary connection, you could say, because it shows that cells aren't producing any more energy than what they need. Okay, so moving on. Okay, so now the reaction is following the formation of fructose 1,6-biphosphate constitutes the energy-producing part of the glycolytic pathway. So here we have cleavage of fructose 1,6-biphosphate yields us with 2 molecules of the 3-carbon sugar glyceraldehyde 3-phosphate, which is then oxidized to 1,3-biphosphoglycerate. Now, the phosphate here has a very high free energy of hydrolysis, so it is used in the next reaction of glycolysis to drive the synthesis of ADP to ATP. So the product of this reaction here is 3-phosphoglycerate, and that is then converted to phosphophenylpyruvate, which I was too lazy to write out. So we have 2 of those molecules, 2 of phosphophenylpyruvate, and this phosphophenylpyruvate is the second high energy contributor intermediate. Okay, so now in hydrolysis of the phosphoenolpyruvates, high energy phosphate bond is converted to pyruvate, but that is coupled with the synthesis of an ATP. So, since we had 2 of those molecules, we've made 4 ATP. 2 are lost in driving the reaction to pyruvate. So we have a net gain of 2 molecules of ATP, and in addition to doing that, this reaction converts 2 molecules of coenzyme NAD to NADH, and that NADH is then used as an electron donor for various other reactions throughout the cell. Alright, so that concludes my video on glycolysis. So, thanks for watching Steven Loves Science. As always, please subscribe, rate, and comment. Alright, thank you for watching this episode of Steven Loves Science.